I had a request to go over the experiment that was amino acid separation. Using ion exchange chromatography. And this is the experiment you do when you want to figure out the sequence of amino acids in your protein. You do this experiment before the actual sequencing experiment with Edmond degradation or the Sanger method. And this experiment tells you which amino acids you have in your protein and how many. And that's important information in general to know. And it really informs and guides you for the sequencing experiment, because the sequencing experiment will involve fragmenting your protein into smaller pieces that you can sequence with Sanger or Edmond. And so you have to know which proteases to use. Because proteases are amino acid specific. And if you find that you don't have a particular amino acid in your protein, or maybe you just don't have very many of them, it wouldn't be a good choice to use a protease that cuts specifically at that amino acid that's not very prevalent. Remember, in sequencing, you want to fragment your protein into small pieces that you could sequence individually, and then you examine the overlap. So the better fragmentation you get, the better your results will be. And I should say that actually it's the proteases you use. Not protease, because you actually technically do several different protease treatments on separate protein samples and then compare the overlaps. So the setup of your experiment is you have a column which is filled with resin with negatively charged beads. As amino acids are eluted from the column, they pass by a reservoir which adds ninhydrin. And I'm going to try to actually draw this just like I did in class so it doesn't confuse anyone. So this is going to be a reservoir of this dye called ninhydrin, which dyes every amino acid that passes by purple, and then whatever passes by will be measured by a spectrophotometer, and the absorbance will be measured at 570 nanometers. And then if you wanted, you could collect your fractions at the end. But what's really key is the data that's going to come off your spectrophotometer. So the absorbance at 570 will be collected over time. And what you're doing over time is that you're washing with solvents of increasing pH. So you start off with your protein that's been hydrolyzed so that you have amino acid. The amino acids are all separated from each other now. And you also have this at a very low pH. So everything's going to be protonated and positive. And everything that you initially pour in will stick to the column, and your amino acids will only elute off when you wash it with a solution that has a pH that matches the pI of that amino acid. Once that amino acid becomes neutral, it doesn't stick to the resin anymore and it elutes. So one of the questions that came up on Piazza is, if everything is purple, if all the amino acids are purple, how do you differentiate between them in your data? And so your data will look like this. Absorbance at 570 over time. And your x-axis could also be labeled the pHs of your buffers, but I think time is more accurate because you'll pour your buffers through and you pour them in a regimented sequence, maybe 1 minute in between each pour. And so then you could just measure everything by time that's coming off of the spectrophotometer. So how you know which peak is what is a great question, and I decided to reveal that to you at the end of my explanation, but I could have also started off with this because this is actually what you would probably do before you run your sample through of your hydrolyzed protein. You do your standards. So you run some standards. So you do the same exact setup. You have your setup just like that. But instead of pouring your hydrolyzed protein through the column, all you do is you pour a pure solution of just one single amino acid. And then you do all your washes and you see where the peak comes out. And you do this for all 20 amino acids. So essentially what you do before you even run your unknown sample, where you get this data, you run a standard set of amino acids to know where the peak will be on the graph. Where, at what point, will your amino acid elute from the column? So this is a standard. You run your standards, you run each of the 20 amino acids, see where the peak goes, and then when you get your data, you compare each of these standards, the location of the peak, to identify which peak is what in your experimental results. So hopefully that cleared up some of the questions. If you have more questions, please do comment or post on Piazza again. All right, thank you.